Our work has really focused on trying to understand how chemicals that we all use and put in the environment can impact on wildlife, but also potentially how it impacts on human health too. My name is Charles Tyler. I'm a professor in ecotoxicology at the University of Exeter. Some of our work established that some chemicals called environmental estrogens can cause feminizing effects in wildlife, but potentially also cause feminizing effects in humans too. And one of the big models that we've been doing in this regard are the transgenic zebrafish. And the beauty about the zebrafish as a model is that it goes through developmental programming during about 4 days, equal to that of about 9 months in a human. And here's an example of a zebrafish, and you can see here's the heart beating and here's the blood flowing. And what we've done in this fish is we've engineered it. We've actually put in a target site for a certain class of chemicals, and we've linked that to a colour that enables us to determine where in the body of this fish it's responding to these chemicals. This has just been exposed to an estrogen, one called Ethinylestradiol. It's found in a contraceptive pill. And what we can see now is this region here, this is largely the liver, and here's the heart valves, which is responding to that estrogen. But if we now switch over to the colour form, and as the fish are exposed to higher concentrations, we see more tissues responding to that estrogen. So this provides us a very intelligent, integrative tool whereby we can start to understand where chemicals are interacting in the body and how they're mediating their effects. These transgenic zebrafish are part of a broader program of work that we're doing, including linking with chemical companies and with pharmaceutical companies like AstraZeneca to better advance the use of models such as the zebrafish for informing on wider health effects, including those in relation to humans as well. And that now and in the future is going to help us reduce the number of mammals that we use, for example, in research and in terms of animal testing. We've also developed new transgenic models looking at multiple interaction sites, and here's one example here. This is a first in terms of here we're looking at the brain of the zebrafish, and here we're looking at 2 interaction sites linked to different colorimetric responses. And we can clearly see here that different parts of the brain are affected by different chemicals. And this type of model provides a real exciting opportunities now to look at interactions of chemicals in our body, because clearly we're not exposed to a single chemical, we're exposed to multiple chemicals. And trying to understand what those multiple chemicals do in terms of health is actually a key and fundamental part of research.